Good morning, children. So today we will continue with the process of translation. Today I'm going to tell you the stepwise mechanism of translation. Before that, I'll tell you the function of ribosomes. Ribosomes the larger subunit and the smaller subunit. What is the function of ribosome? Ribosomes have two functions. The first function of ribosome is decoding the transcribed message. Decoding the transcribed message. So this message is transcribed where? This message is transcribed in messenger RNA. So this messenger RNA which have the transcribed message, it is decoded by ribosome. That is, translation is brought about. At the same time, the larger subunit of ribosome, the larger subunit the larger subunit has the enzyme peptidyltransferase, and this enzyme peptidyltransferase is used for the formation of peptide bond of polypeptide chain. The second thing is termination of polypeptide chain. So these are the functions of ribosomes. Yesterday I told you the structure of ribosomes also, and these are the function. First, the transcribed message is decoded, and second, the larger subunit of ribosome, which has the enzyme peptidyltransferase, it performs these two functions. Okay, now we come to stepwise mechanism of translation. Before this, I'll rub this for you so that I can start with the process of translation. You can write what is translation? A process which takes place in cytoplasm, during which the coded genetic message on messenger RNA polypeptide chain. So the coded message which is on messenger RNA, it is converted into polypeptide chain. This process is known as translation. Now, the detailed mechanism of translation, it goes on like this. The first step in the mechanism of translation is activation of amino acids. See, for the process of translation to continue, amino acids are required because what is formed as a result of translation is polypeptide chain or a protein molecule. As a result of translation, what is formed? A polypeptide chain or a protein molecule. This is formed as a result of translation. Now, what is this polypeptide? Whether we say polypeptide chain or we say protein molecule, it's the same thing, because what is protein molecule, what is polypeptide, it's same thing. And many amino acids connected by peptide bonds. Many amino acids connected by peptide bond, bond, peptide bond, peptide bond. Many amino acids connected by peptide bond. This is one polypeptide chain. Many peptides are there. Polypeptide chain. Or protein. What is protein? Many amino acids connected by peptide bonds is a protein. It's the same thing. So as a result of translation, protein molecule or polypeptide chain is formed. Now, that means what do we require? We require amino acids. For making a protein molecule or a polypeptide chain, we need amino acids. And where are these amino acids present? These amino acids are present in amino acid pool, which is there in the cytoplasm. The cytoplasm of the cell has amino acid pool where amino acids are stored. And from there, the amino acids are brought by transfer RNA or T. This is a cell, this is the nucleus, this is messenger RNA, and here the ribosome has attached. Okay, now this is the nucleus. Now suppose this is the amino acid pool on the cytoplasm. Amino acids are there. So the amino acids are required here so that this chain or a protein molecule can be formed, which will go from the exit side, which will move out of the exit side. Now the tRNAs from here will bring the amino acids from the amino acid pool to the site of translation, which is ribosomes. So it will bring the tRNAs here, and these polypeptide chain will be formed, which will come out of the exit site, and we will get a polypeptide chain. So what is the first raw material which we need for the process of translation? These are the amino acids. And one thing more, the amino acids which are present here, these are not in activated form. These are in inactive form. So they are to be activated first. First of all, the amino acids which are brought by tRNA, these amino acids which are there in the amino acid pools are to be activated. First, they are to be activated, and then the transfer RNA must be charged with amino acid. That means they should carry amino acids on them, and then they will come to the site of protein synthesis. So this happens. Now, the mechanism of translation, the first thing which I write here is the machinery. Machinery, I have already told, I think, what do we need? Anybody who remembers what I told regarding the machinery of translation? Machinery of translation, what do we need for translation? Yes, what is the machinery for translation, beta? What is the machinery of translation, bache? Anybody who remembers the machinery of translation? Yes, bache? Anybody who remembers the machinery? Yes, messenger RNA, tRNA, ribosomes, amino acids. See, if amino acids will not be there, then it won't take place. The translation will not take place. So machinery which is required, machinery for translation. The machinery for translation consists of tRNA, very important. And if the message is not there, what will they code for? Messenger RNA, another important thing. If protein factories will not be there, then where the protein will be synthesized? So our ribosome is very important. Peptides, protein molecules are synthesized by amino acids. So amino acids are very important. If amino acids will not be there, from where how the protein molecule will be formed? Then one enzyme which is very important is amino acyl tRNA synthetase. Amino acyl tRNA synthetase, this enzyme. This is the machinery for translation which is required. See, tRNA is required because it gets charged by the amino acid. That means it carries amino acid from the amino acid pool to the site of protein synthesis. Messenger RNA, it has got the instructions that which particular amino acid has to join where. All the coded messages there in the messenger RNA. Ribosomes, because ribosomes are the protein factories. It receives the transfer RNA. Then peptidyl transferase, which is present in the larger subunit of ribosome, forms the peptide bond. If peptide bond will not be formed, a protein molecule cannot be formed. So that is also very important. Then amino acids, because proteins are formed by amino acids. So amino acids are very important. Then there is this one enzyme, amino acyl tRNA synthetase. This is very important enzyme which is required for activation of amino acid as well as charging of tRNA. So let's come to the first step of the mechanism of translation. The first step during the mechanism of translation is activation of amino acid. See, I told you the amino acids are present in the amino acid pool of cytoplasm, and there they are present in the inactivated form. They are not activated, they are present in the inactivated form. Now, what happens? These amino acids, they have to be activated. And how do they get activated? They get activated when they combine with ATP. See, always for activation, energy is required. So these amino acids which are present in the amino acid pool on the cytoplasm, they get activated when they react with ATP in the presence of this enzyme, amino acyl tRNA synthetase. So when amino acids, they react with or they combine with ATP in the presence of this enzyme, a complex is formed. I'll write that reaction for you. The reaction is amino acids. I'm writing in short, amino acids plus ATP. Then in the presence of the enzyme, amino acyl tRNA synthetase in the presence of this enzyme. Or we can just write down here E, because such big name, it is difficult to write when we form the complex. So I'll write just E. Fine, for this amino acyl tRNA synthetase, I'll write E. So when it reacts, when amino acids reacts with ATP in the presence of enzyme, what is formed is amino acid. Now, when it reacts, that means ATP will break. Hydrolysis of ATP will take place, and this ATP will break to form the monophosphate. So it will form ATP gets converted into AMP with the release of energy plus the enzyme. This also combines with it, and this complex we get this complex when this amino acid reacts with ATP in the presence of enzyme. We get amino acid AMP, adenine monophosphate enzyme complex. This plus, because see, PP, this was triphosphate, and we are getting here monophosphate with the release of energy. So along with this, pyrophosphates are formed. So we can also write it PP. So PP is formed. So this is what happens during the activation of an amino acid. During the activation of amino acid, where are these amino acids? Amino acids are there in the amino acid pool in the cell. That means if this is a cell, this is the cell, this is amino acid pool. Here in amino acid pool on the cytoplasm, the amino acids are there. All amino acids are lying. But these are in inactivated form. Inactivated form. So for translation, these are to be activated. And how do they get activated? By this reaction. By this reaction, the amino acids which are there in the amino acid pool, they get activated and form amino acid AMP enzyme complex. This is formed. Amino acid AMP enzyme complex is formed. Now, very important thing, this reaction in the presence of this enzyme is catalyzed only in the presence of magnesium ions. So Mg2 plus, Mg, I'll write here Mg2 plus, presence of Mg2 plus is very important to catalyze this reaction. This reaction takes place in the presence of Mg2 plus, magnesium ions. Yesterday also I told you importance of magnesium ions in one of the combinations, in one of the reactions. Can you tell me if you remember? Yesterday I told you magnesium was required somewhere else, if you remember. And in the absence of magnesium, that process cannot take place. Where was it required? If anybody remembers yesterday's, what I taught? Yes, beta. Yesterday also I taught you a process. Yes, small and large subunits joining. Yeah, yes, that's very good. Paras, very good. Because this is very important that what I teach a day before, you should remember. Sukh Sahaj. Yes, beta. Sukh Sahaj, good. Joining of small and large subunits in ribosomes. Yes, I told you that the smaller subunits and the larger subunits, they lie like that in the cytoplasm. They lie like that. They are not joined. They join only at the time of translation. So when translation takes place in the presence of magnesium ions, these two subunits, the smaller subunits joins with the larger subunit, forming a complete ribosome unit. So this also takes place in the presence of magnesium ion. Now this is another thing where this magnesium is required, that is to catalyze the reaction in the presence of this enzyme, which is amino acyl tRNA synthetase. So this way the amino acids which are there in the amino acid pool, they get activated. Now the second, now they have got activated. So the second step comes, which is charging of tRNA. Charging of tRNA. See, in the cell, in the amino acid pool, the amino acids have got activated. Now these amino acids are to be carried from this amino acid pool to the site of translation. Suppose this is the ribosome where the translation is taking place. This is the ribosome where the translation is taking place. So these are to be taken from the amino acid pool to the site of translation. How they are carried to the site of translation? I told you a substance which acts as pulley for carrying the amino acids from the amino acid pool to the site of translation are transfer RNA. So transfer RNA are to be charged with those activated amino acids. That means they should carry activated amino acids on them. That is when the transfer RNA carries activated amino acids from the amino acid pool, we call it charging of tRNA. So the second thing which takes place is charging of tRNA. During this, what happens? This amino acid AMP enzyme complex, which is formed as a result of activation of amino acid, this joins with the binding site of specific, this complex which is formed, amino acid AMP enzyme complex. Where will it bind? It will bind with the active site of tRNA. Now, what is that active site of tRNA? If anybody remembers, when I told you the structure of tRNA, I told you this is the site where the amino acid attaches. What is that site? Anybody remembers? Who will tell me what is that site? Binding site of tRNA where the activated amino acids bind? A site. It has got some other name, beta. Yes, Paras, Suksahaj. Any other name it has got? Yes, bache. Yes. Yes, bache. Acceptor site. Acceptor site is in ribosomes, beta. No, acceptor site is in ribosome, and that accepts the amino acid. I'm saying that if this is transfer RNA, I'm saying what I want to tell you. What I am asking is, if this is transfer RNA, what is the active site where the amino acid will get bind? What is that? What is that site, binding site on this transfer RNA? Yes, this is a transfer RNA which I have drawn. Now, can you tell me what is that binding site on this transfer RNA? Yes. Now, if you remember, binding site on this transfer RNA, amino acid attaching site. Any other name it has got, Paras? Any other name? It has got any other name? Anticodon loop. Tanish, anticodon loop is here. Anticodon loop is here, the last one, which connects to the codon of messenger RNA. Anticodon is the lower loop. If you remember, bache, the amino acids, they get attached here. They get attached here. This is 3' side, 3'. It has a hydroxyl group. It is 3' side, which has a hydroxyl group, and this is the amino acid attaching site on transfer RNA. So here the amino acids are attached, 3' site. Here the amino acids get attached with this functional group. And which part of the amino acid gets attached? Which part? By which side it gets attached to this 3' end? If you remember the structure of amino acid, it has two functional groups. Yes, by which side? By which site it gets attached, amino acid, to this 3' OH site where this hydroxyl group is present. By which side does it get attached? Anybody, if you remember? So, yes, Paras. It is COOH site. If you remember the structure of amino acid, it has, this is the group, this is CH3, here we have NH2 amino group, and this side we have COH. So from this side, it gets attached to the transfer RNA. Okay, and to that side, amino acid attaching side, which is 3' end, which has hydroxyl group, where hydroxyl group is present. Here the amino acid gets attached. And which type of amino acid? The activated amino acid. When this activated amino acid gets attached to transfer RNA, this type of transfer RNA is said to be a charged transfer RNA. When, I repeat, when activated amino acid gets attached to 3' end where hydroxyl group is present, 3' end by its COOH side, when it gets attached to transfer RNA, such RNA which carries a activated amino acid, it's said to be a charged transfer RNA. This is said to be a charge transfer RNA. So what happens during this reaction? During this, again, this reaction is again catalyzed in the presence of same enzyme. See, in this process, just one enzyme is used, and that is amino acyl tRNA synthetase, which I have written there on the board in the machinery. This enzyme again catalyzes this second step also, which is charging of tRNA. Now what happens? During this, now what do we have? We have this complex. So amino acid AMP enzyme complex. This is what we have. This, it will combine with transfer RNA for charging. It will combine with transfer RNA. And as a result of combination of this complex with transfer RNA, what we are going to get is Again, the same enzyme. Okay, same enzyme will catalyze the reaction. That means amino acyl tRNA synthetase. The same enzyme will catalyze the reaction, and as a result of this reaction, what we will get is that this tRNA will get bonded, will get binded to amino acid complex, amino acid. We can write only amino acid also, and this will be released and this will be released. They will get separated. So AMP is released plus enzyme, which is of no use now. This is also released. And this is the second reaction which takes place during this reaction. What happens? The amino acid AMP enzyme complex joins with the binding site of its specific tRNA. Then as a result of this, what happens? The tRNA gets charged. AMP and enzymes are, the enzyme is finally released. And what happens? See, when ATP was converted into AMP, energy was released. When ATP was converted into AMP, energy was released. And that energy is retained in this complex. That energy which was released, that energy is retained in tRNA amino acid complex. We can use the word complex also here because it is a complex. So that energy which was released during the conversion of ATP to AMP, that energy is retained in tRNA amino acid complex, and this energy later on is utilized for the formation of peptide bond in a polypeptide chain. For the formation of peptide bond in a polypeptide chain. So this is the second step which takes place, which is charging of tRNA. The third step is another important step during the process of translation, and this is activation of ribosomes. As I told you, ribosomes consist of two subunits, and both the subunits, the smaller and the larger subunit, they lie on the cytoplasm. They are not connected with each other. They join or they attach with each other only during the process of translation. During the process of translation only, the larger and the smaller subunits, they attach with each other. Otherwise, they lie on the cytoplasm like that. So as the onset of translation starts, the magnesium ions in the presence of, again, I'm repeating it, in the presence of magnesium ions, the smaller subunits attach to the larger subunits and form a complete ribosomal complex. A complete ribosome is formed when ribosomes are formed when the larger and the smaller subunits they attach with each other in the presence of magnesium ions. Now, what is the function of both the subunits? See, magnesium ions are required. Magnesium ions joins or attaches the smaller and the larger subunits. The smaller and the larger subunit. This takes place in the presence of magnesium ion. Now, what is the function of the smaller and the larger subunit? The smaller subunit attaches to the messenger RNA. Attaches to messenger RNA. And what is the function of larger subunit? The larger subunit has the enzyme peptidyl transferase. Peptidyl transferase, which carries about two functions. Number one is formation of, which carries out two functions. Number one is formation of peptide bonds. And the second function which is carries is termination of polypeptide chain. So this is the third step which takes place during the process of the translation, when activation of ribosome takes place in the presence of the magnesium ions. Okay, and what is activation? Larger subunit attaching with a smaller subunit of ribosomes in the presence of magnesium ion. So this is the third step, which is very important during the process of translation. We come to the fourth step, that is initiation of polypeptide chain. Now the fourth step begins, and the fourth step is initiation of this is the fourth step, initiation of polypeptide chain. Now, what happens during the initiation of polypeptide chain? Now, again,